Mart, excellent introduction. Thank you very much. I will continue and probably repeat a little bit some of the things that you already have mentioned, but now focus really on CDNF and try to explain a little bit further why we believe that the CDNF has significant potential in prevention of dopamine neuron degeneration and stimulation of neuronal regeneration. So as Mart already briefly mentioned, the current standard of care in Parkinson's is lacking. Basically, we're currently treating symptoms, and we've been doing so for the past 50 years in a more or less similar way, based on dopamine replacement. There's for decades there's been attempts, and in the past 10-20 years, increasingly so, attempts to develop therapies that could actually slow down, delay, stop, or even reverse partially the progression of this chronic disease. And as such, growth factors like and neurotrophic factors like GDNF, nutrient, CDNF offer exceptional potential for this. They do have challenges, of course, being proteins, but Mart already mentioned a few strategies which can be used to capitalize their molecular properties to support neuronal survival, functionality, and improve also in long term or provide protection against disease progression in Parkinson's patients. And for the past decade or so, even longer, there's been a series of preclinical studies that have provided evidence that support CDNF's actions, particularly in Parkinson's, possibly in other degenerative CNS diseases as well. And this TreatER project provides another important milestone in CDNF development, as we have now established safety of the intracranial CDNF in Parkinson's patients. So the study that will present some of the results today met its primary endpoint and as such supports further development of CDNF in Parkinson's. I come a little bit back to the topic that Mart already introduced a little bit. So we have multiple aging-related chronic degenerative diseases such as Parkinson's, Alzheimer's, frontotemporal dementia, ALS, even ischemic stroke, where we have substantial neuronal cell death occurring in the brain for various reasons. And because the adult human brain is very limited in its capacity to generate new neurons to replace the dead neurons, the strategy should be to rescue those neurons that actually are still alive but have lost their functional phenotype and could be rejuvenated in a way to support the remaining functions. And not only are neurons very long-lived, they're also highly polarized in their structure. The cell bodies of dopamine neurons that degenerate in Parkinson's, located in the substantia nigra, are located very far from the axonal terminals in the regions of putamen, for example. A good comparison would be that if the cell body of a dopamine neuron would be the capital of Finland, Helsinki, the distant synaptic terminals would be located in the furthest parts of Finland up north in the Lapland. So this is a very, very big challenge for the neurons to maintain their functionality and survival. Moreover, there are various types of stress, like oxidative stress, inflammatory stress, endoplasmic reticulum stress, unfolded protein stress. These types of stresses that make it even more difficult for neurons. And we know that the neurons are particularly sensitive to disruptions in proteostasis. I will come back to this in a couple of slides. So what happens in these diseases is that the neurons don't die overnight. This is a slow progress. So first we start losing the synaptic terminals, then the axon becomes functionally impaired and could actually retract, we could still have the cell bodies present and alive. This is the time window where therapies, neurorestorative therapies like CDNF, should be most functional and most efficacious. So if we miss this time window, it means that the neurons actually have been lost. And in order to replace these, no restorative therapy such as a neurotrophic factor would be efficacious. We would need to actually bring new neurons into these brain regions. For example, stem cell therapies, some embryonic tissue graft therapies, experimental therapies that have been tested in Lund, for example, are then the solution. But of course, this is probably not a functional solution for millions of Parkinson's patients worldwide. So in Parkinson's, several cellular mechanisms important for functionality and survival are impaired. One such important organelle in the cells is the mitochondrion, the power plant of the cell. Another one is lysosome, which is a recycling factory or recycling plant of the cell. And then we have the endoplasmic reticulum that's highlighted here. It is a very complex cellular organelle involved in many cellular functions like protein synthesis, folding and secretion, lipid synthesis and metabolism, calcium metabolism, and so on. And also importantly, the ER serves as an important sensor of unfolded protein stress in the cells. So now we come back to proteostasis. So proteostasis basically refers to a balance between all the proteins in the cell. So in a mammalian cell, we have at least 40 million protein molecules in a very tightly packed space. And proteins are very sensitive. Their function depends on their three-dimensional structure. So both the synthesis and folding are important parts of the proteostasis network. And then the third part, the degradation part, is equally important. And we know that in many chronic degenerative neuronal diseases or central nervous diseases, we have problems with proteostasis. We have accumulation of oligomeric, misfolded, aggregated proteins that actually have cause multiple problems in the tissue. They are problematic for the cells to deal with, but they can also induce inflammation, which is a known counterpart in many of these neurodegenerative conditions. And CDNF actually can modulate the proteostasis situation at multiple levels, and particularly does so by interacting with the number of proteins inside the endoplasmic reticulum. So in Parkinson's, the proteostasis disruption is actually can be seen as a common neuropathological finding known as Lewy bodies. So Lewy bodies do not exist in young, healthy brain, but when people age, these type of protein junk starts to accumulate in the brain and sometimes lead to a disease. And in Parkinson's and in some related synucleinopathies, these Lewy body type structures, as shown in different morphologies here, accumulate in the brain. And these Lewy bodies are mostly composed of a protein called alpha-synuclein. And this is a very important therapeutic target now in the field of Parkinson's. So here we see on the top left-hand side, we see some smaller Lewy body structures inside a cell. On the bottom left, large inclusions. And then on the right-hand side, we see dystrophic neurites, which are filled with aggregated alpha-synuclein. And this causes big problems for the neurons and contributes to the degeneration. So CDNF was originally discovered by Martarma and colleagues at the University of Helsinki a little over 10 years ago. And originally it was named as a neurotrophic factor, conserved dopamine neurotrophic factor. However, a little over 10 years of research, very intensive research by Mart and some other laboratories have shown that actually CDNF is quite distinct among the neurotrophic factor family of proteins. So here's the three-dimensional structure also shown by Mart. So this gave the first insight in how CDNF may be different from the other known neurotrophic factors. So actually, importantly, in biology, structure dictates function. Proteins are the building blocks of the cells, but also responsible for a majority of the functions in the cells. And each protein falls in a unique three-dimensional structure, which is in a way critical for its functionality. And here on the right-hand side, you can see the structures of a GDNF and a related molecule called neuterin. And you can see that their structure is actually very similar. They're composed of two monomers, one in blue, one in orange, identical protein molecules that are held together with the covalent disulfide bridge. Also, they have very similar, highly similar secondary structural elements shown as these kind of like arrows here, so-called beta sheets. But when you now compare this to CDNF, although the overall shape may look a little bit similar, this is, from the molecular perspective, this is anything but similar. All the secondary structure elements are distinct. The functional unit is a monomer. It's composed of two independently folded domains, and it has very different structural motifs also, sequence motifs. So this now we're every year learning more and more about the mechanism of action of CDNF, but it looks like it's indeed entirely different from the classical growth factor concept. So growth factors like GDNF bind to specific cell surface receptors and activate secondary signaling cascades that eventually promote cell survival. CDNF interacts with intracellular targets inside the endoplasmic reticulum and regulates cellular proteostasis. And also, CDNF can protect the cells against alpha-synuclein toxicity and pathology, while GDNF doesn't seem to have this property. Moreover, because of certain sequence motifs present in GDNF and neuterin, their diffusion in the brain tissue is quite limited. On the bottom here, you see images from a non-human primate study where GDNF and CDNF were infused into the brain, and the white area here shows the volume of diffusion. For GDNF, it's significantly more limited compared to CDNF. And of course, the bigger the volume of diffusion, the more target cells in the brain can be reached. So this could be an important proxy for efficacy. In summary, CDNF affects multiple disease-relevant mechanisms in cells. So as mentioned, the endoplasmic reticulum is a key target of CDNF. The unfolded protein response pathway there inside the endoplasmic reticulum is an important target, and particularly for maintenance of proteostasis, supporting the functionality of the cells, this is a key target. The number 2 here illustrates activation of protein kinase B, so-called AKT, which is important also for neuronal survival. Then the number 3 illustrates CDNF's effects against alpha-synuclein toxicity, oligomerization and toxicity. And these are all intracellular effects inside neurons. And then we have number 4 illustrating here an important component, which is the neuroinflammation. So it is known that CDNF can also suppress production and secretion of pro-inflammatory cytokines by glial cells, the immune cells of the brain, which play a very important role in supporting the disease process. And finally, the effects of CDNF on tissue level are actually surprisingly long-lasting. We know based on preclinical studies that a single administration of CDNF can actually have effects that last for weeks, up to a month, if not longer. And this is very important now, thinking about the approach that has been taken in the clinical study. It's actually important to keep in mind that always more, sometimes less is more. So in a way, intermittent modulation of these target mechanisms is believed to be better for the brain tissue than continuous presence of CDNF protein in high levels. For example, other approaches that would result in constant high levels of CDNF would be continuous infusion of the protein, or for example, gene therapy. The problem here is that with gene therapy, for example, we cannot turn off. If we find that there's an adverse event, we could not turn off the expression. This would be, in the worst case, could be a catastrophe for the subject. So we believe that the intermittent dosing is really key for CDNF, the harnessing the therapeutic effects of CDNF. And in this clinical study, we have used an intracranial approach. And because CDNF is a protein, it doesn't penetrate the blood-brain barrier. And as you may remember, our target cells are located deep inside the brain. The cell body is in the substantia nigra, and the axonal projections and synaptic terminals in the brain area called putamen. And the putamen here now is the most important target region of the brain. And in this study, we used intracranial administration by neurosurgically implanted catheters to make sure that CDNF can be delivered exactly to this brain region. And actually, we have tested this similar approach in experimental models of Parkinson's. So these are, I'll show you just briefly some data from a non-human primate study that was actually carried out by Mark Sarma and Judy Cameron from the University of Helsinki and University of Pittsburgh, respectively, several years ago. And here in this model, the animals are lesioned with a toxin called MPTP, which fairly selectively kills and injures the dopamine neurons. The little inset here shows a brain section from a healthy animal. And all the dark staining here illustrates dopamine neurons. It reveals dopamine neurons, their cell bodies, and their thin fibers traveling from the substantia nigra towards the striatum. And now when the MPTP toxin is given to the cells, there's a very significant loss of these neurons. You can see that there's the cell bodies are reduced in number, and also the fiber density is very much reduced. Now, we started intracranial treatment with CDNF in these animals 6 weeks after this lesion was established. And as you can see here, after 3 monthly infusions of CDNF, we have a remarkable neurorestorative effect. We have increased number of cell bodies, the little puncta here, and also significantly increased density of dopaminergic fibers, as you can see the thin lines going here and there. And this is really a proof of concept for this type of neurorestorative effect following a dopaminergic neuron injury. Importantly, when these animals were tested for their motor functions, CDNF after 3 monthly infusions improved the motor functions by about 50%. So this is the monkey equivalent for the human UPDRS. So you can see here that the red balloons and the blue balloons, the red ones being CDNF and the blue ones being GDNF, both improved quite remarkably in the study, while the placebo group here, the gray balloons, didn't really show much improvement in their motor symptoms. So this very similar approach is now translated from preclinical models to humans in this TreatER clinical study. In summary, in Parkinson's, dopamine neurons die slowly, and treatments like CDNF have in various preclinical studies shown significant therapeutic potential, particularly in functionally recovering the degenerating neurons and also the neural functions. Optimally, a neurorestorative treatment like CDNF would be started very soon after the onset of symptoms. You may remember from Mark's slides that after 3, 4, 5 years into the disease, the dopaminergic innervation in the brain is significantly reduced. So the further the disease progresses, the less there are neurons left to protect, and the effects are expected to become weaker. Alpha-synuclein pathology lies at the core of Parkinson's and appears to be an important mediator also for disease progression. CDNF improves cellular proteostasis in general and protects neurons from alpha-synuclein toxicity and may modulate progression of alpha-synuclein pathology as well. And this is, of course, again, slightly different as compared to some other neurotrophic factors like GDNF, for example. Based on CDNF pharmacology and mode of action, intermittent protein therapy offers several benefits over continuous infusion or gene therapy, despite the challenging intracranial route of administration. Thank you for your attention. We have now a couple of minutes left for questions. And I believe that there actually are no questions so far. So please remember that we have also other opportunities to ask questions. You can use the chat box in the control panel of the webinar app, and you can ask questions after each session or at the end of the panel discussion.